नमस्कार। आप सभी लोग अपने अपने घरों पर सकुशल होंगे। सो स्टूडेंट्स, लेट्स मूव टू अवर नेक्स्ट टॉपिक। द टॉपिक नंबर 2, that is the cell cycle and cell divisions. Its second topic, that is the meiosis. In previous we have already talked about the cell division, that is known as the mitosis, in which we have talked about the different stages, how the chromosomes can be divided, and the different stages of this mitosis can be followed, that is the karyokinesis, cytokinesis, and the karyokinesis, in which we have talked about the prophase, in which early and late prophase, metaphase, anaphase, and the telophase. After that, it can be followed by the cytokinesis, means the cell division of the cytoplasm, especially in the animal cell and in the plant cell. So let's begin to start our next topic, that is the meiosis. First of all, we have to know that the meiosis is the phenomenon which can be occurs in life cycle of the involved the process of sexual reproduction. Means generally the basic difference between the animal mitosis and meiosis, that is the mitosis can be always take place in the process of the somatic cell divisions, right? But this meiosis phenomenon, this meiosis process will always take place or involved for the sexual reproductions in that conditions. We can say the productions of the offsprings by the sexual reproductions involve the fusion of two gametes, right? Mean each gamete has a complete haploid set of chromosomes, right? Or n number of chromosomes. We can say the gametes are formed in the male and female reproductive organs by the meiotic cell division. Means those gametes which can be used for the sexual reproduction, they can be always help to develop with the help of this meiosis or meiotic cell division. We can say it is a sliced form of the cell division which can be reduces the chromosome number in haploid condition. Suppose we can say the 46 number of chromosomes in the human beings, but during the gametogenesis, during the formation of gametes, they will become, each gametes can be contain 23 chromosomes, right? So we can say when the daughter nuclei receives only one set of the father and one set from the mother. We can say they can be always, they will get and receive the chromosomes from the maternal or the paternal. So we can say its results to produce the haploid daughter cells, and in the meiosis the nucleus divides twice, but the replication chromosome take place once, especially in the interphase of the meiosis first. So students, you must take care that there is a special thing about this meiosis cell division, that in the meiosis cell division the nucleus divides twice. nucleus जो है दो बार divide होता है, but the replication of the chromosome that will take once, right? Especially in the interphase of the meiosis first, right? So we can say, so this meiosis division is also known as reductional division, but on the other hand the mitosis is known as equitational division. So we can say, especially in the case of the diploid organisms, meiosis take place during the formation of the spores, gametes, where the haploid organism will take place during the termination of the zygote, and so on, right? So there are some special features of the meiosis are found over there. So they are the, generally we can say that meiosis undergoes two successive cycles of the nuclear and the cytoplasmic division, that is meiosis first and meiosis second. Evenly we can say there are some other thing, there are some other characters are that we can say the meiosis first and second occur one after another and with the very short or the number of interphase, means there is not too long gap between the meiosis first and second one. Evenly we can say after the replication of the parental chromosomes, initiations of the meiosis first take place in order to the production or identical sister chromatids in S phase stages. Evenly the meiosis take place about the day to get complete instead of the hours. Generally this meiosis can be needed to complete it within one day at least for this process, right? But mitosis can be completed within the hours or some minutes. On the other hand, we can say the pairing of chromosomes or recombination take place during the meiosis. the pairing means we can say the pairing of homologous, same kinds of chromosomes can be pairs together, right? That is called pairing of homologous chromosomes. Recombination take place during the meiosis, and evenly we can say after the second meiotic cell division, 4 haploid cells are formed, right? But in case of the mitosis, there is only 2, where the somatic parts can be divided. But in case of the meiosis process, there is 4 haploid cells are formed over there. So you must pay attention about this one. Then we have to directly come to our topic, that is the meiosis first. Generally, meiosis first can show some special division under the condition of the different stages, that is prophase first, metaphase first, anaphase first, and the telophase first. So first of all, we have to know about this one, that during the meiosis first division, the homologous chromosome of each pair separated from each other, and they can reach to separate the daughter cells, with thereby reducing the number of chromosomes from the diploid to haploid. Means that especially in the meiosis, first of all, the diploid chromosome can be changed into the haploid. We directly in which we can say the 2n change into the n form, and that is known as heterotropic or heterotypic division also. Evenly, as I have told you, that this further divided into 4 phases, especially prophase, metaphase, anaphase, and telophase. Right? So first of all, we have to know about this prophase first stages. In that particular conditions, we have to know that in the prophase first, the most complicated and prolonged phase of this cell division, right, in the mitosis. Means generally it is also compared to the mitosis. Evenly we can say, especially in the prophase first stages, they get subdivided into 5 subphases. Some more division, this prophase first can be again divided into 5 sub-stages or subphases. They are, as you can see in the diagram, that is liptotin, zygotin, pectitin, and the diplotin, and the last one is known as dikinesis. Means we can say, you all must know that during the prophase first stage there are some 5 subdivisions are also found over there, that is the liptotin, zygotin, pectitin, diplotin, and dikinesis. These are the different 5 subdivisions of the prophase first stage or prophase first division. So let's begin to this prophase first. First of all, we have to know about this liptotin stages. As you can see in the diagram, right? You can just see in the diagrams the chromosome where the thin thread-like condensations. Means all the chromosomes are just like a thread-like structures over there. We can say it is the first stage of the meiotic division, right? In this particular condition, the different features which we can say, especially as I told you, that generally the chromosome become gradually visible under the light microscope. Evenly, the centrioles start moving towards the opposite ends or the poles, and each centriole develop astral rays. Means generally in the same conditions as the first you have seen, as we all know, there are the centrioles are found over there. As you all know, I think that the structure of centriole, that such structures, they will start to move from here to their poles, like here. Means there they will start to form over there, right? And these, after reaching the poles, they will start to form the astral rays inside the nucleus, like this one, right? Or you can say the chromatids are closely adhered together. Means generally these fibers like structure can be seen in that particular conditions in which they can be found over there. Evenly we can say each chromosome is attached to the nuclear envelope through the attachment of the plate at the both of its ends. Generally they will started to attach their plates to the nuclear envelope also, right? Or we can say the chromosomes can be double constructing to chromatid due to the duplication process की वजह से. After that there is another process that is called zygotin. Means we can say zygotin take place after the completion of the previous ones, This is also short-lived stage during the liptotin, right? Or we can say, especially in the zygote, the homologous chromosomes pair up जो same kinds of chromosomes होंगे. As you can see in the diagram, the blue color and the red color. The red one are the homologous and the blue color are the homologous. So this way you can say that both are the homologous chromosomes are pair up. This pairing is done such a way that the gene of the same characters present on the two chromosomes lies exactly opposite to each other, and this process is known as the synapsis. So you can just think about this one. As you can see over there, the homologous chromosomes come to lie side by side, right? And that pairing is known as synapsis over there. Evenly, we can say the reverse in the zygote, it is revealed from the electron micrographic studies that can be formation of synaptonemal complex take place by the pair of homologous chromosomes that show synapsis, and the complex so form account of the synapsis from the bivalent and tetrad. Means you can just see the particular structure, they can be formed such structures over there, that is bivalent or the tetrad structure can be found over there. But how? Because the space have highly specialized fibrils, organelles, synaptonemal complex, they can capable to form over there. So this structure can be found over there, and due to this they can be maybe diet. As you can see, generally the chromosome with 2 chromatids called diet, chromosome with 4 homologous, they are known as tetrad, and the 2 chromatids of the same chromosomes are known as sister chromatids in the different ways. Or in the last we can say those 2 homologous chromosomes is called non-sisters chromatids. Evenly the crossing over occurs in that particular conditions, but these stages are found in the Pecatan stage. Evenly we can just understand about this tetrad and the bivalent in that condition of the zygote. After that can be start to follow to another stage, that is called Pecatan. The word itself say Pecatan. We can say during the zygote, where the pairs of the chromosomes become twisted spirally around each other and cannot be distinguished separately. जो Pecatan stage में zygote के बाद they can be twisted from each other and they cannot be easily separated from each other. So we can say that each other and cannot be distinguished from each other those chromosomes. So that stage is comparatively long lived as compared to the previous 2 stages. Means generally the Pecatan stage is for a long durations in compared to the liptotin and the zygote. Only the different changes which can be seen, especially in the Pecatan, they are, we can say that in that particular condition the bivalent chromosome clearly seen as tetrad. जो बाइवलेंट क्रोमोसोम से उनका जो स्ट्रक्चर है वो आसानी से टेट्राड के फॉर्मेशन में दिखाई देता है, यानी 4 के 4 जुड़े हुए दिखाई देने लगते हैं. साथ ही द एक्सचेंज ऑफ जीन्स और क्रॉसिंग ओवर बिटवीन द 2 नॉन सिस्टर क्रोमेटिड्स of the homologous chromosomes occurs, means especially during the Pecatan the crossing over stages can be also found over there. So in that particular time we can say the recombination nodules are formed over there, which can be appears at intervals of the synaptonemal complex. By the end of the Pecatan, recombination gets completed, leaving the chromosomes linked as the sites of the chromosome over. Also, you all must know about the crossing over, because in that condition whenever they were crossing over will form. What about this crossing over? Generally the exchange of the genetic material take place between the non-sister chromatids or the 2 homologous chromosomes. Pay attention that the exchange of the genetic material take place between the non-sister chromatids of 2 homologous chromosomes is called crossing over. Generally you can say it is the final lead to the recombination of genetic material on the 2 chromosomes, means they can capable to exchange the genes, the genetic materials. So that process is known as the crossing over. Then we have to come on the next stage, 4th stage, that is known as diplotin. Means especially in the diplotin, what will happen over there? this is the longest duration. We can say this diplotin stage is the longest duration in all of this prophase first stages. We can say especially in this the septonumeral complex appears to get dissolved while the chromatids of each tetrad remains clearly visible. जो सेप्टोन्यूमरल कॉम्प्लेक्स था इट कैन बी डिजोल्व, और उसके डिजोल्व होते क्या होता है जो क्रोमेटिड्स से जो टेट्राइड स्ट्रक्चर में थे वो इजीली विजिबल हो जाते हैं. एंड ड्यू टू दैट वी कैन से the recombined homologous chromosomes of the bivalent get separated and form chiasma, X-shaped structure. Right? Means they can capable to form the chiasma. Chiasma क्या होता है बेटा? जनरली जो हमारे क्रोमोसोम होते हैं, राइट, तो जब ये दूसरे क्रोमोसोम के साथ ये यहाँ पर एक्स शेप स्ट्रक्चर बनाएंगे. यू जस्ट सीन सपोज this is a chromosome, right, and there is another chromosome like this, and when this arm can be formed like this, right. So you can just see, especially in that particular condition, right, this X shape structure over there is this one especially. This is known as the chiasma, right? Means they can be just crossing. So we can say the recombined homologous chromosomes of the bivalent get separated and form chiasma or chiasmata, that is X-shaped structure seen in the diagram easily, right? And we can say that this chiasmata formation is necessary for the separation of the homologous chromosome which have undergone the process of crossing over. If there is such process will not take place, तो क्रॉसिंग ओवर की जो प्रोसेस के समय जो हमारा होमोलोगस क्रोमोसोम सेपरेशन हुए थे उसके लिए ये प्रोसेस इट इज वेरी नेसेसरी ओवर देयर, राइट. सो आफ्टर दैट क्रॉसिंग ओवर और आफ्टर दैट chiasmata formations, that is structures, we can say that chiasmata formation is necessary for the separation of the homologous chromosomes which have undergone the process of crossing over. जो क्रॉसिंग ओवर के टाइम पे ये प्रोसेस हो रही होती है, इसलिए उसके लिए chiasmata फॉर्मेशन का होना बहुत जरूरी रहता है. आफ्टर दैट वी कैन से स्पेशली the another and the last stage of the prophase first, that is the dikinesis. Means what will happen in the dikinesis? As you can show, generally in the dikinesis the chromosome condense again into short thick rhodolet. Means generally in the dikinesis process there is another thing. Directly you can say that this process is also known as terminalization, and due to this, to the shifting of the chiasmata toward the end of the chromosomes, and during this particular conditions the chromosome become fully condensed and nucleus degenerates and breakdown of nuclear envelope is into vesicle occurs, and also the formation of meiotic spindles in order to prepare the homologous chromosomes for the separation also occurs. मीन्स इसकी पर्टिकुलर कंडीशन में सेपरेशन कराने के लिए स्पिंडल का फॉर्मेशन हो जाता है, जैसे कि आपका माइटोसिस में भी होता है, राइट. एंड ड्यू टू दैट द होमोलोगस क्रोमोसोम्स टू बी सेपरेटेड आल्सो अकर्स, एंड वी कैन से दैट डाइकिनेसिस स्टेजेस रिप्रेजेंट्स द ट्रांजिशंस फ्रॉम द प्रोफेज टू द मेटाफेज स्टेज. मीन डाइफेज स्टेज जो है ये क्या करती है, ये इट कैन आल्सो हेल्प टू शो द changes or transfer from the prophase to the metaphase stage in that particular condition. So this process can be take place in that particular time. After that the stage of the metaphase will come. As you can see during the metaphase stage, right, they can be just same to the mitosis like as in the mitosis. So we can directly say the bivalent during this phase arrange themselves on the two parallel equatorial plates. Means they can be started to come on the parallel stages. You can just see as in diagram, they will just come at the plate over there, and the centromere project little bit towards the periphery since there are two centromeres in each bivalence. Such a centromere is joined to the chromosomal fibers, or we can say the fiber of the homologous chromosomes are ऑलवेज इन द अपोजिट डायरेक्शन में, जो क्रोमोसोमल रहते होमोलोगस क्रोमोसोमल के अपोजिट डायरेक्शन में हो जाते हैं. इवनली वी कैन से इन दैट पर्टिकुलर कंडीशंस ड्यूरिंग द एनाफेज फर्स्ट, the in which we can say the homologous chromosomes break into connection with each other and get separated. Generally they can be with the help of spindles, Estrell's rays, they can attach with the gonadotroph and they can started to separate it out from that particular conditions. As you can see, right, in our previous stage you can say that these are the aligned over here, but in that condition after in the anaphase first you can just see that these chromosomes centromere can attach over there, they can be separated from here and they can be separated over here. Means you can say this division, this furrow-like structure will start to develop over there. So we can say, especially in the anaphase first, that the separation of the homologous chromosomes is known as disjunctions, and the separated chromosome are univalent and are also called dyads. Means dyads क्यों? आपने देखा इसमें टेट्राड्स था, 4 थे, राइट, बट इन दिस कंडीशन यू कैन जस्ट सी स्पेशली दैट ट्वाइस, 2 हो गए, सो दे आर ऑल्सो नोन एज द डाइट्स. एंड ऑन द रीचिंग ऑफ द एनाफेज द 2 group of chromosome are produced which are heavily half number of the chromosome, and after that they can be formed the 2 structures over there and they will form that the नंबर ऑफ क्रोमोसोम बिकम्स द हाफ. पहले में 14 थे, 4 थे, अब इसमें कितने हो गए? 2 हो गए. सो एट द स्टेजेस ऑफ द टेलोफेज स्टेज फर्स्ट, मीन्स यू कैन जस्ट सी इन द टेलोफेज स्टेजेस ओवर देयर. सो स्पेशली इन द टेलोफेज स्टेजेस वी कैन से द क्रोमेटिड्स at each pole of spindle usually remains uncoiled and get elongated. Means generally in the same way यहाँ पे भी जो क्रोमोसोम्स आपके पहुँचे थे, जो क्रोमेटिड्स पहुँचे थे, वो क्या होंगे? स्पिंडल की मदद से वो अनकॉइल्ड, दे कैन रिमेन अनकॉइल्ड एंड गेट इलोंगेटेड. अनकॉइल्ड होंगे, जो आपस में ट्विस्टेड थे, खुल जाएंगे और खुलने के बाद क्या होंगे? दे कैन बी गेट इलोंगेट, लंबे हो जाते हैं, राइट. सो वी कैन से दैट होमोलोगस क्रोमोसोम्स रीच एट देयर रेस्पेक्टिव पोल्स, अलग अलग पोल्स पर चले जाएंगे एंड रीअपीयरेंस ऑफ द न्यूक्लियर मेम्ब्रेन एंड द न्यूक्लियस टेक प्लेस. तो दोबारा से वहाँ पर न्यूक्लियर मेम्ब्रेन बन जाएगी और जो न्यूक्लियस का डेवलपमेंट हो जाएगा न्यूक्लियस का. आफ्टर दैट इट कैन बी स्टेज ड्यूरिंग द साइटोकाइनेसिस कैन बी फॉलोज एंड द साइटोप्लाज्म एंड द अदर ऑर्गेनेल्स डिवाइड इनटू द 2 equal halves of the cell. So after that the interkinesis is another stages. We can say that this stage can between the 2 meiotic division. Means जो meiosis फर्स्ट और meiosis सेकेंड है उसके बीच में भी एक डिवीजन होता है, that is called interkinesis after the cytokinesis, right. In this particular stage this can be found between the meiosis first and meiosis second. It is generally shorter lived. During this process no replication of DNA occurs. It is necessary to bring 2 haploid DNA in daughter cells. So in fact conditions in such structure can be found over there, right. And after that the another process that can known as meiosis second will start, but we have to not talk this time, we have to talk about this meiosis second in next time. So students, you must learn up to this stages, right, and you must completely just remind about the meiosis first, that can be followed the 4 stages: prophase, metaphase, anaphase. Especially in the prophase first I have told you it is sub stages, 5 sub stages are found over there: liptotin, zygotin, pectin, diplotin and dikinesis, in which the total development, total homologous chromosomes and their chromatids can be take place, right, and it can be followed with the help of the completely up to the telophase stages. And after cytokinesis and the interkinesis can be take place due to which the 2 which can be always found between the meiosis first and second. So student, up to here we have just completed the whole stages of this. So you must know that in our next topic, in our next lecture, we will talk about the meiosis second. So after that we can capable to complete this chapter. So student, you can prepare your notes.